Hello, everyone. Hello. Thanks for finding your way to Osgood East and the rest of the AMR Symposium, the beginning of the rest of the AMR Symposium sessions. I'm Natasha Christie Holmes. I'm the Director of Strategy and Partnerships for EPIC, the Emerging and Pandemic Infections Consortium. We've very happily been a partner on this symposium for the last 3 years. We're excited to be back. And this year presented an interesting opportunity, a great opportunity to align the last of a 3-part speaker series that we've been hosting in conjunction with Institute for Pandemics and the Temerty Centre for AI in Research and Medicine, T-CAIREM and IFP. So we're really excited today to have a speaker series about AI and infectious disease, specifically about drug development and antimicrobial development. And I'm going to hand it over to my colleague Mamatha Bhatt, who's a hepatologist and clinician scientist and the co-lead of the Transplant AI Initiative at Toronto General Hospital. She's also Associate Professor of Gastroenterology and Hepatology at the University of Toronto, and she's the Partnerships and Engagement Lead for T-CAIREM, and she'll introduce the rest of the session.
Well, good morning, everyone. It's my great pleasure to welcome you to today's exciting panel on accelerating discoveries in biology and medicine using AI. This is the third and final panel discussion co-hosted by EPIC, the Institute for Pandemics, and T-CAIREM. Quick mention, this discussion is CPD accredited for physicians. You can scan a QR code and we'll show at the end of the discussion how to fill out the evaluation form. I wanted to also invite you all to become members of T-CAIREM. It's free and it's now the largest AI in medicine network across Canada. You can visit tcairem.utoronto.ca to learn more. We're delighted to have Ivan Semeniuk as today's moderator. Ivan is an award-winning journalist who reports on science for Canada's national newspaper, The Globe and Mail. Ivan's beat ranges from health and the environment to the frontiers of physics and the role of scientific research in Canadian society. Prior to joining the Globe and Mail in 2013, he was chief correspondent for Nature, based in Washington, U.S. bureau chief with New Scientist magazine, and a producer and columnist with the Discovery Channel. Thank you so much for joining us, Ivan, and I'll invite you to come to the stage and then introduce our panelists for today.
Thank you very much.
Oh.
There was a force field there for a moment. Let me check the logistics first. We have our keynote. So, everyone, welcome to the panel. It's wonderful to see you. This is the third in a series of panels, and I can tell you they've all just been unequivocally exciting and eye-opening as a sort of introduction to a very rapidly changing field, actually a rapidly changing sequence of fields where AI is having a big impact, and we can see that there will be more impact to come. So we have, as we have done these panels in the previous iterations, we'll have a keynote speaker, and then we have 2 additional panelists who are sort of respondents to this, and then we will also be throwing open to questions, both for the people here in the room and to those who are following online. So I want to encourage you right now, as I do to all of my students when I teach journalism courses, to have your questions ready now. Never be in a situation where you don't have a question, because this will be your opportunity to ask to your heart's content about things you want to know about as they come out of our conversation today. I just want to check. We're going to bring up our keynote speaker first, and then I will introduce our responding panelists after that. So now I will turn to our keynote speaker, and who is César de la Fuente. He is a presidential assistant professor in the Department of Chemistry at the University of Pennsylvania, where he leads the machine biology group. His pioneering efforts in machine-aided discovery include the development of the first computer-designed antibiotic with efficacy in animal models. Pretty interesting. His lab has also been at the vanguard of developing computational methods for proteome mining, including, for the first time, exploring the human proteome as a source of antibiotics, and finding therapeutic molecules in extinct organisms I find this very cool. In the process, he's launched the field of molecular de-extinction. Dr. de la Fuente received his PhD at the University of British Columbia and pursued his postdoctoral research at MIT. He's received numerous international awards. He's also an elected fellow of the American Institute for Medical and Biological Engineering. There's quite a bit more I could say, but instead, in the interest of time, how about we now welcome our keynote speaker today, Dr. César de la Fuente. Thank you.
Well, thank you so much, Ivan, for that very kind introduction. Yeah, it's really great to be back in Canada, what was my home for a long time. And so, yeah, it's great to be here. So today I thought I would tell you a little bit about what we've been doing the last 5-6 years, applying AI methods and computational methods for antibiotic discovery. So I'll take you a little bit through this journey. In terms of the learning objectives for the lecture, I hope to convey that antibiotic resistance is a huge global health problem that we all really should care about. Second, that using AI and computational methods, we are now able to accelerate antibiotic discovery, and I'll briefly mention a couple of projects where we've been able to do this. Number 3, I'll introduce the concept of molecular de-extinction and some of the work that we've been doing in this emerging area. And finally, I'll mention how AI can also be used to sample through the entire tree of life to try to find new antibiotic molecules. But let's start from the beginning. Well, I'm sure this audience doesn't need much introduction here, but antibiotic-resistant infections are really a huge global health problem. They affect every corner of the world. They're currently associated with 5 million deaths per year. And the projection is that by 2050, that number is going to increase to 10 million deaths per year around the world. If you do a quick calculation, that's about 1 death every 3 seconds. So I think it's really imperative that we try to come up with out-of-the-box approaches to try to discover new antibiotics. So I'll just tell you about what we've been doing since I arrived at the University of Pennsylvania. When I first got there, I basically formulated what I think is a fundamental question, which is, can computers be used to accelerate antibiotic discovery? And what we propose is that instead of using traditional methods for coming up with new antibiotics, which is basically a very physical process, right? You go around nature and you take soil samples or water samples and you try to purify active compounds that might be present in all of that organic matter. As you can imagine, that's a very, it's a process reliant on trial and error and very painstaking process that at the end of the day might not give you any promising candidates. So instead of doing that, we propose to take advantage of the decades worth of biological data or biological information that we have at our disposal in the form of genomes, proteomes, and metagenomes that have been sequenced over the last decades. And we propose to come up with algorithms that could sort through all this amazing biological data to see if we could find encoded or hidden within all this data potential molecules that could be antibiotics. So sort of the analogy is like using a magnifying glass to browse through all the biological information that we have in the world to see if we can find molecules that could be useful. The other motivation for using computers to accelerate discovery is that it takes a long time, like I mentioned, to come up with new molecules, and it's a very expensive process, right? So you can imagine that if we come up with algorithms that can speed up this process, we could reduce the time and the cost associated with coming up with new antibiotics. So that was the hypothesis that we formulated now 6 years ago. And so I'm going to tell you briefly how we did this chronologically. We started by taking inspiration from image and speech recognition algorithms, but instead of recognizing faces or sounds, we wanted to recognize molecular patterns that were present in biological data, like I mentioned. We started very simply by mining individual proteins to see if we could find within the protein antibiotics encoded or hidden within the protein structure. So a protein we can visualize in 3 dimensions. We can also display it in 2 dimensions. And what the algorithms do is they run through this code and they identify regions that are predicted to be antibiotics. And these are given by these different colors here. So it's only a prediction, right? But it's pretty powerful because it allows us to explore individual proteins and identify within the protein fragments that might become potential antibiotics. And then using synthetic biology tools, we can extract those fragments to try to develop them into potential new medicine. All right, so we did this for a couple of projects. Then with advances in compute power, we realized that we could scale up. And so we wondered whether instead of just mining individual proteins, we could do entire proteomes. And the proteome is all the proteins encoded in a genome. And so we embarked on this journey to try to mine the human proteome as a source of antibiotics. This imposed a huge scaling challenge because we're going from one protein at a time to suddenly 42,000 proteins. And so we had to come up with an algorithm that was sufficiently inexpensive, computationally speaking, to be able to sort through all of our genetic data, all of our genetic code, to see if we can find new molecules. To our surprise, the algorithm that we developed, it was this pattern recognition algorithm. It was able to do the job in just 1 hour, which was quite amazing to see, with relatively little compute power that we had at the time. And this very simple algorithm, it enabled us to sample all our genetic data and identify thousands of new molecules that we call encrypted peptides that were previously unrecognized to have antimicrobial properties in our body and perhaps play a role in the immune system. What we then did is we actually synthesized out of these thousands that we discovered, we synthesized dozens of them. We can use chemistry to actually make these molecules in the lab. And we tested them against bacteria in Petri dishes, but also in preclinical mouse models. And I won't go into all the data, but we saw that when we treated infected mice with some of these small peptides, they were able to reduce the infection significantly in these mice. So this work, it sparked a lot of new ideas in my lab, one of which was the fact that we're finding these thousands of new previously unrecognized molecules in our genome, in our proteome. It probably means that they're not just there. So we came up with this new idea where we thought that we might be able to find similar molecules all throughout evolution and throughout the tree of life. And since we had centered our efforts on looking at the human proteome first, we decided to see whether we could find similar compounds encoded in our closest ancestors. And our closest ancestors are Neanderthals and Denisovans. And so in the process of thinking about all of this, we came up with a framework for discovering molecules that we call molecular de-extinction, like Ivan introduced. And the whole purpose of this framework, this conceptual framework, is to really identify or discover molecules all throughout evolution and throughout time, to then be able to synthesize them, to be able to learn how changes that occurred on those molecules throughout evolution influence their biological function. Their biological function might be antibiotic properties, it might also be immunomodulatory properties, anti-cancer properties. And so we're now really using this conceptual framework for thinking about all of this. Okay, going back to archaic humans, so to be able to tackle that problem of whether we could find antibiotics in ancient humans, we had to develop a new machine learning model that at a very high level, what it does is it takes all the proteins from Neanderthals, from Denisovans, it chews them up in little fragments, and then we run 2 different filters, one of which is a machine learning filter, the other one of which is a human expert filter, to predict whether those fragments will be antimicrobial or not. And I think it's important to highlight that every decision that we make in my lab, we take into account the recommendation made by the algorithm, but also the recommendation made by the human experts in my lab, the scientists. And I think it's quite telling of what I think the future of science is going to be. It's going to be, I think, a collaboration between machines and humans. And I don't subscribe to the camp of machines taking completely over. I think humans, we still are useful, to a large extent. But anyway, this algorithm, it allowed us to identify small therapeutic peptides in ancient proteins. Here we have some examples in Neanderthals and Denisovans. We use the modern human proteome as a control for our experiments. But this is only a prediction, right? The algorithm predicts that these things are going to be great antibiotics. But then in my lab, we always do experimental validation. We always synthesize everything, and we test it in the lab, right? So that's what we did here. We synthesized a lot of these small proteins that the computer identified, and And we tested them in vitro and also in preclinical mouse models. Just quickly, I'll show you just the preclinical data in 2 different mouse models. This is a skin abscess infection model on the top, and on the bottom, we have a deep thigh infection model, which is typically the one that the Food and Drug Administration in the US wants to see for exciting preclinical candidates. I won't go through all this data. This is published work, but just to say that the top hit was Neanderthalin. And another thing I haven't mentioned is that we had to come up with new names for all these compounds that we're finding. So the convention that we're using is that we name them based on the organism that produces them, right? So Neanderthalin comes from Neanderthals and so on. But this work demonstrated that molecular de-extinction was a feasible framework. It also identified these molecules in ancient humans, and it gave us the courage to ask a more perhaps ambitious question, which was, why not just sample every extinct organism known to science? But once again, this imposed a scaling challenge. We're going from 3 organisms here, modern humans, Neanderthals, and Denisovans, to now having to mine hundreds of organisms. So we had to develop a more powerful machine learning model. And so this is a multi-year project in my lab, but we developed a new AI model, and we trained a new AI model from scratch. We call it Apex. This is the architecture that Apex works under. And the really interesting thing in terms of its application is that Apex, it essentially opened a window into the past. It enabled us to sample all organisms all throughout evolutionary history, including the Holocene and the Pleistocene, as we can see here. And here I'm just showing representative organisms where we found some of the top antibiotic candidates. It includes this penguin that was extinct in the 50s, not so long ago, magnolia trees that disappeared throughout evolution. It includes the woolly mammoth, ancient elephants, giant sloths, and many, many other creatures from the past. So essentially, using AI, we've been able to sort through all this extinct data and identify new antibiotics that could be useful today. To give you a sense of the scale of this exploration, here is all the organisms that we sampled. Many of the molecules that we found, we then synthesized and validated experimentally. Obviously, I'm not going to go into all of this, but this is just to give you a sense of the magnitude of the exploration. And then we tested them in preclinical mouse models, once again, of infection. Just to summarize some of the results, the best compounds came from the woolly mammoth, a molecule that we're calling mammuthicin, and from the ancient sea cow, the giant sloth, and the extinct giant elk. And the really interesting thing is that in this case, using Apex, we've been able to identify molecules that have anti-infective properties that are comparable to polymyxin B, which is a last resort antibiotic used in hospitals worldwide. So it was really exciting to see that we could actually find things that are promising in mouse models. So we published this paper recently. The journal Nature Biomedical Engineering highlighted this new concept of looking at extinct organisms as a source of antibiotics and perhaps other useful molecules. So why molecular de-extinction? I'm just going to tell you some things that we've learned so far. It has already enabled us to discover an entirely new sequence space. So if you visualize all the proteins and peptides that have been discovered through science up to date, and then you compare them to what we've discovered by looking at ancient organisms, many of the small peptides that we found in ancient organisms, they fall outside of the prototypical sequence space. We think that by comparing molecules throughout evolution, we can unlock new biological insights. We can learn about how they evolved and why they evolved in certain ways, and why certain mutations or level of conservation influence how they change over time. I think this has already expanded the way we think about molecular diversity and what might be possible in terms of sequence space of molecules. The hypothesis that we have is that a lot of these things that we're finding, they might be playing a role in the immune system throughout evolution. And we're currently testing this hypothesis in my lab. And finally, we believe that by bringing back molecules from the past, this can help us address present-day problems. So as you can imagine, contemporary pathogens that are killing hundreds of thousands or millions of people today most likely have never seen molecules that come from thousands or hundreds of thousands of years ago. So we're essentially bringing back an entire new arsenal to try to combat present-day pathogens that are giving us a lot of problems today. A couple of ramifications from a lot of this work that I thought I would highlight. One on the bioethical side. As you can imagine, when we were initially uncovering a lot of these antibiotics from ancient organisms, it was very exciting scientifically. But then I started worrying a little bit, is it okay for us to synthesize this in the lab, these molecules? Because when we do multiple sequence alignments, for a lot of the things that we found, we don't find them in the biological world around us today, in living biology. And so is it okay for us to bring them back through chemistry? And so we've been consulting with bioethicists to make sure that we innovate, but that we do so responsibly. And second, on the patenting side, I thought I would mention that when I went to the patent office to ask, basically I told them that I knew that natural sequences are not patentable because they belong to humanity. But then I asked them, what about these ancient sequences that we're finding that are no longer in biology, in the natural world? Are those patentable? And basically, this is interesting because it's opening up a whole new area of patent law. And it's quite entertaining to see lawyers fight with each other about whether we should be patenting some of these things or not. So I thought I would mention that too. In the meantime, I've teamed up with my colleague Andrew, and we've written this opinion piece for Nature Biotech on some of these concepts and our thoughts, our 2 cents on some of these ideas. In terms of next steps, we plan to mine the world's biological information. We think of biology as an information source. If you think about it, biology is just a bunch of code, nucleotides, amino acids, and if we can develop the right algorithms, we can sort through all this amazing amount of code, and we can try to find new antibiotics and new, potentially other types of useful therapeutics. We're not Google, so we can't do this in 3 months, but what we're doing is we're sorting through the whole tree of life. And since we first initially focused on eukaryotes in the form of modern and ancient humans, we next decided to go after another huge branch of the tree of life, which is bacteria. So we decided to sample the huge diversity of the microbial world to see if we could find antibiotics encoded or produced by these amazing little creatures. And I'm going to tell you just briefly about 2 collaborations that we just completed. We published these 2 papers this past summer. The first one, we decided to use machine learning algorithms to explore what we call the global microbiome. These are almost 90,000 microbial genomes and over 60,000 metagenomes that come from that come from all around our planet, from many, many different habitats, including soils, oceans, corals, even pets like cats and dogs, the oral cavity. They're really an amazing representation of the microbial diversity on our planet. And by using an algorithm, we're able to sort through all this amazing data and identify almost 1 million new antibiotics. So remember, it's really hard to come up with one. Here, we were able to identify almost 1 million. We've made this, we've called this AMP Sphere. This is a website. We've made all of this information publicly available, open access for everyone to be able to really look at these sequences, be able to maybe synthesize them and be able to characterize them. So hopefully amongst all of us as a scientific community, we can, the dream is that we can maybe take some of these forward and they can eventually help people. We validated a lot of these in preclinical mouse models as well, by the way. This is the second collaboration with our colleagues at Stanford, where we decided to look at human microbiomes as a source of antibiotics. So human microbiomes are good bacteria that live in and on us. For example, a lot of them live in our intestines. And we thought this would be a great environment for coming up with new antibiotics, because if you think about it, these microbes, there are billions of them living in a relatively limited space. So they have to really fight each other off, right, to take over a particular niche and then to grow into little cities and little countries, microbial countries. So we thought that this would be a great environment for coming up with maybe new compounds that individual microbes use to fight each other off in sort of like a chemical warfare. And by sampling almost 2,000 human microbiomes, we're able to identify individual peptide molecules that individual microbes produce that they use to fight off microbes in their vicinity, in their surrounding environments. So again, we think this is all part of a microbial warfare that is happening. And we tested them in preclinical mouse models. The top hit that we found, we call it Prevotellin 2. It comes from the gut microbe, Prevotella copri, which we all have in our intestines. So again, another reminder of the incredible capabilities of microbes to come up with new chemistries and to come up with smart solutions to problems that they face, right, in their daily lives. So just to sort of to summarize, like I mentioned, with traditional antibiotic discovery methods, it can take a lot more than it takes to complete a PhD program to come up with exciting candidates. Today, my lab, in a few hours, we can come up with hundreds of thousands of candidates, and then it takes a little bit longer to test them, but not a super long time. Within a week or a couple of weeks, we can do that. And so, really, for all the hype that AI has, I think this is I consider this a success in the sense that it's enabled us to dramatically accelerate antibiotic discovery. And this is just a sample of some of the compounds that we've been able to test all the way to preclinical mouse models of infection at the moment. And we hope to continue to grow this list with the ultimate dream of hopefully some of this or derivatives thereof hopefully can help people in the future. And with that, I'd like to thank lab members, past and present, our amazing collaborators, funders, and I'll just leave you with links to website, email, and X handle in case anybody wants to get in touch. And once again, thank you so much for having me, and would love to take questions during the panel discussion.
César, why don't you have a seat right at the far end? That's terrific. It's very exciting stuff. I'd now like to bring up our additional panelists, starting with Dionne Aleman. She's Associate Dean of Cross-Disciplinary Programs in the Faculty of Applied Science and Engineering at the University of Toronto. Her research focuses on the application of operations research to healthcare systems to improve the quality, timeliness, and efficiency of care. And I know her work involves optimization, simulations, graph theory, and machine learning, among other things. Thank you very much, Dionne, for joining us. Thank you very much. Finally, also, I'd like to welcome Artem Babaian, who is a assistant professor of molecular genetics with the University of Toronto's Terence Donnelly Centre for Cellular and Biomolecular Research, where his work includes among other things, his research goals and quests includes exploring the total biodiversity of Earth's RNA viruses, and as we were discussing recently, exploring entities that are virus-like but not quite viruses. So exciting new domains of discovery ahead. So thank you very much for both of our panelists, Artem and Dionne. I guess I'm going to move over here as well. So I'm going to, again, encourage everyone to think of your questions. I'm going to just start the ball rolling here with a quick discussion with the panel, but we're going to go to questions as quickly as we can to make the most use of time. And I will be able to draw questions from online from the podium as well. So for those of you who are joining us online, please send in your questions as well. And I do want to remind everyone, quick reminder, for symposium attendees, I think both in the room and online, if you are participating in the symposium's online game today, you receive points by asking questions to our panelists via the live stream chat app or here in the room. And as a journalist, I approve of the idea of rewarding people for asking questions. So you can gain points by asking questions today as well. I'd like to ask César a question, then I'm going to turn to our panelists. César, you mentioned towards the end of your presentation, we're not Google, you're not Google. And of course, next month, in a few weeks, we're actually going to see the Nobel Prize in Chemistry awarded to researchers who used AI methods to crack the problem of protein folding, the AlphaFold. Mm-hmm. They were resourced by Google to some extent, which obviously accelerated. They had a number of other things going for them for that particular problem. But it does make me wonder, given the scope and the significance of antimicrobial resistance as a problem in the world, which you laid out so well, is it time for some kind of larger institutional, global, concerted effort to make use of these tools in a bigger way to tackle that problem, or is it too soon? What do you think is needed here to really get to the heart of it?
Yeah, I think that's a great question. Thank you, Ivan. Yeah, so I mean, AlphaFold is the software that was designed by Google to predict the structure of proteins, right? Giving a particular sequence of amino acids, what's the 3D structure of that particular sequence. But AlphaFold, so this AI model, it was trained on the protein data bank, which is public data, right? That thousands of people have collected over decades. So that was a concerted effort, probably billion, probably, I don't know how much money it costs, but tons of money. And it was basically an organized effort to be able to generate such a data set that is invaluable, right? Which was able to then train AlphaFold, and for AlphaFold to be successful, that's what it used. So I think that's a great question. Is it time for a Manhattan level type of project for antimicrobial resistance, either in Canada or worldwide, where we can all share data, right? About antibiotic resistance microbes and so on, so that then we can build models like AlphaFold for antimicrobial resistance, for surveillance purposes, to predict the spread of resistance in particular populations, to discover new antibiotics, which is what we're doing. But all that sort of data, you can apply it for many different applications. And given the magnitude of the problem of the AMR problem, maybe it is time for a Manhattan project for AMR.
Interesting. Artem, I'd like to ask you, I mean, you're working in the virus world, but I think more importantly, sort of trying to exploit these new computational tools as they're coming online. How would you characterize how things are changing, how fast things are changing, or how researchers even can stay on top of these tools to get the most out of them?
I like to kind of draw a parallel that in the 1970s, like with the advent of PCR and restriction enzymes, we saw biologists become chemists, right? And it gave birth to this kind of molecular revolution in biology. I think we're at exactly the same paradigm today, where we're seeing computing unlocking a new world of genetic information. Because I think, as César mentioned, like the heart of genetics is information, right? Even you go back to the central dogma, it's not talking about molecules, it's talking about the flow of transfer of information. And computers are information systems. And so now what we have to do is we have to learn a new language of computational exploration, like César has done fantastically, to mine the available genetic sequence data that's available. To give you a perspective of how much data we're talking about today, so the public database of sequence data, it's called the Sequence Read Archive. This is the collection of sequencing data from everybody in the world, accumulated over the past 15 years. It's been doubling every 2 years exponentially, faster than Moore's Law. Contains over 28 million samples, or 80 petabases of sequencing data. So that's an 8 followed by 16 zeros. My kind of favorite statistic about this is if you were to generate that volume of data as like a Manhattan project, you would have to run your Illumina machine just to generate the data for $3.7 to $14.9 billion. So you add in sample acquisition and personnel costs. What we have today, before even starting this project, is a $50 billion genetics resource that we as a community have collectively built. So the real question is, how do we as biologists scale up to this kind of new era of petabase scale sequence analysis? How do we mine the entire world of genetic information?
Right. And I guess that has implications going all the way down to even training and teaching and all of those things. So let's go further downstream. Dionne, you're dealing with kind of more of the clinical setting, the healthcare setting, the hospital setting. We're still not there yet with these AI-guided discoveries, but there's a sense, I think, people have of that pipeline. We're kind of looking upstream to that pipeline. I just want to ask you I guess I'm asking you to look in the crystal ball a little bit. What do you foresee as that pipeline begins to emerge in more of a clinical setting? What are the considerations? How might it affect health for everyone? By the time that starts to affect people.
Yeah, well, César mentioned towards the end of his talk talking about gut biomes, and that's a very hot area of research, trying to figure out how do people's gut biomes relate to this symptom, this disease, that affliction. And I could imagine, at some future point, that, tests will be developed, whether it's, blood tests or, fecal tests or, something that will allow physicians to understand what's happening in an individual patient's gut biome that might be causing them health problems, and then using discoveries like what César has presented to create, say, a little customized probiotic supplement pill. Here, take one of these every morning, and we'll see you in a month, and you'll be cured, right? I mean, obviously, that's jumping the gun, maybe many, many decades, but you can imagine that something like that would be coming down the pipeline and will require sort of several stages of AI and machine learning before we get to that implementation phase of an individual doctor making a decision for an individual patient.
Got it. One more question for César, and then again, I encourage for people who are in the room, we have microphones right there. If you've got a question, come on up. I'll turn to you next. We do have some questions from online as well, so I'll be able to go to those in a moment. So come on up. Don't be shy. Come to the microphones. I'm sure you have questions. César, we live in a time now, I think, where AI is mentioned every other sentence. You get your cereal box and you're having breakfast, and on the cereal box it says, now with AI. I'm just kidding, but you get the idea. So it feels like we're living in this world of equal measure surprise and skepticism. It's the astonishing things are happening in the AI space as applied to healthcare and biomedical discovery. But still, I think some skepticism is warranted in terms of how far are we from this affecting people directly. How should we think about this in terms of separating sort of the hype from the real promise?
I think I agree that there's certainly a lot of hype. I think these machine learning methods can be useful tools to achieve things like in the protein structure prediction field, or to try to come up with new antibiotics, but we're still very far from seeing a medicine that we can take that has been generated or designed by AI. And in fact, I think we were mentioning that yesterday, but there are a lot of drug discovery companies using AI as their primary engine to come up with new medicines, and none of them have been successful yet. So I think we're still not quite there with at a number of levels, but I think it has been, and I think it is a useful tool if you have the right amount of data and the right level of data that you can use to train these algorithms. But I think data generation is actually a crucial part of it, and we're still not, in many, many domains, we're still not quite there in terms of having sufficient amount of data to train these algorithms properly.
Got it. All about the data. Excellent. Well, we've got some questions, so why don't we go here? I'll start, and I will also go to some questions online as well, just looking at the time to see where we're at. So that's good. I will start here on my left. You've got a question.
Thanks very much. Just a question for César, and maybe the other panelists can comment as it raises an issue that's probably apropos of this discussion. Are your models mature enough to employ them in a generative way? Have you learned enough about antimicrobial peptides to generate new ones never seen before?
Yeah, great question. We have a number of projects now where we're using large language models and generative AI tools to come up with entirely new things that are different from what we find in biological sequence space. So we are working on that at the moment. We also have another project where we're combining deep learning with Bayesian optimization, so also using generative AI approaches for optimizing existing things that we've already found. So, and we've seen some success there. I think I was just going over a paper yesterday where giving a particular template that we give it out of the many, many things that we found, these algorithms that we're developing now, they 72% of the time they improve upon the template. They modify the template and they improve it. So I think, and that's experimental data. So I think certainly we're now venturing into generative AI and other approaches as well.
Thanks.
Got it. How about a question on this side?
So first, César, I'd like to endorse the fact that we don't have enough data. Computers, of course, do better when they have a lot of data to feed in at one end. But I'd just like to mention another possibility that I think is really important, and that is that drug development is a continuum. It doesn't just involve identifying a molecule, but that molecule has to have the right pharmacokinetic, pharmacodynamic, toxicity, et cetera, properties that allow it to be developed into a drug. And you showed a picture where you have to discover 10,000 drugs to get one good one. So the issue is, can we now start to use computational means, especially artificial intelligence, to start to predict all those other things that make a successful drug? And there's where we need a lot more data, but I think we have this possibility, especially with multi-layered neural networks, where you basically have multiple inputs, so you can feed in things like sequence, the actual pharmacokinetic properties of the drugs, the toxicity properties of the drugs, the solubility properties of the drug, et cetera, and then out the other end, you can come up with each of these things optimized, so we can start to find molecules optimized not just for activity, but for all the activities that make a successful drug.
Yeah, thanks, Bob, for that great question. I mean, I couldn't agree more. I think to make a drug like Bob is mentioning is a multi-objective optimization problem. It's not only about optimizing for antimicrobial activity, but to actually create something that people can take. You need to make sure it's not toxic. You need to make sure it's sufficiently stable for it to exert its function, right, to kill bacteria before it gets degraded. Pharmacokinetic, pharmacodynamics, how it's distributed throughout the body to actually kill the bacteria that we want to kill. So we're very slowly making progress in all those areas. So the thing is that we have to generate data first. So we have different projects in the lab where we have a cytotoxicity project, a hemolysis project, a stability project, and we're very slowly and painstakingly generating the data that then we will use to train the algorithms. Our ultimate goal is in probably several years, we'll be able to build individual models for each of those parameters and then combine them in a super algorithm that will be able to do multi-parameter optimization. So then you will be able to say, predict a particular sequence of amino acids that is a great antibiotic. It doesn't kill good bacteria. It doesn't kill human cells. It doesn't kill red blood cells. It's sufficiently stable. That's the ultimate goal where it's slow. I mean, just to give you an example, Apex, the deep learning model, so that's a project that we started in 2019 to generate the data, right? And we weren't sure it was gonna work. And what we did is after, I think 1 year and then 2 year, 3 year mark, we tried to train Apex on the data that we had, but for a while it was not sufficient data. So the performance metrics of the algorithm were just not good. And it took us, I think, 3.5 years to have sufficient data for when we saw that Apex was actually good at predicting function from sequence. So it was, and thank God it worked, because otherwise my team would have killed me, right? Because for a while I'm like, no, we need to generate this data to create this algorithm, and they were like, sure, and it was not working for a long time. So now to put it into context, it's pretty stressful, right? Because you're betting on something and you're not sure if it's going to work, and then eventually it did work, thankfully. But it was a lot of work in the back end to build the data sets, and that demonstrated to me and to my team that it's possible to do it if you generate good data using standardized conditions. That's what we did, using the same media, the same inoculum, the same pH, everything. So now we're trying to do that with all these other parameters, and so that's the goal. Thanks, Bob.
Interesting. Good questions. How about, let me go one online, and then I will go back to the microphone. And we still have we still have time for people to come to the microphone. Here's a couple from online that I'll combine together. Interesting questions from my point of view. First of all, how did you and your team obtain material for sequencing from these long extinct animals? And also, something I was wondering as well, are fungal organisms being assessed for antimicrobial potential? It's kind of an obvious question.
Yeah, so like it usually happens in science, we stand in the shoulders of giants. There were many scientists over many decades that came up with sequencing methods for archaic DNA, which is very hard because you don't have a lot of DNA. And so in order to amplify, you have to develop specific methods to do that. And actually what people have done is they've amplified mitochondrial DNA instead of chromosomal DNA, because we have a lot more copies of mitochondria. So we are actually it's easier to amplify mitochondrial DNA. Perhaps the pinnacle of that field was 2 years ago, I think, when Svante Pääbo was awarded the Nobel Prize in Medicine for developing sequencing methods for archaic DNA. What this means for us is that a lot of these data are available digitally now. Mm-hmm. And so what we've done is develop algorithms to be able to mine those data. We are looking at fungi, we're looking at viruses, we're looking at plants, we're looking at essentially the whole tree of life. Yeah, archaea.
Interesting.
As a source of new molecules, yeah.
Excellent. Let's go to another question at the microphone.
very, very inspiring work. I'm wondering, because of the issues with peptides and pharmacokinetic properties and so on, whether you can use this essentially chemical space that you've been exploring, as peptides are chemicals, to have access to the druggable chemical space of smaller molecules. That is, can you make small molecule mimetics of the chemistry that inhibits bacterial growth?
Yeah, thanks for that question. We are growing the chemistry team now because what we're doing is we're taking a lot of these templates and we are tweaking them chemically through cyclizations, disulfide bonds, stapling. And so we are now we're also looking at peptidomimetics, at conjugating them with sugars, with lipids to increase the PK/PD profiles. So we are now growing the chemistry side of the lab to be able to take this to the next level.
But how about translating it to the small molecule space, like that 10 to the 60th molecule space that people talk about?
We're doing a little bit of that, conjugating some of these. So some of these peptides we have a project where some of these peptides can penetrate into bacterial cells, right? So we're trying to conjugate them with small molecule antibiotics that cannot penetrate, let's say, into a gram-negative cell to see if we can do that. Computationally, we're also looking at the non-canonical sequence space to see if we can explore that to incorporate non-canonical chemistries into what we're doing. Thank you.
I'm going to go online for a couple more questions. Again, I'm going to combine two questions. This kind of goes a little bit further beyond the substance of today's talk, but just also looking, I'm sure all of you are part of conversations like this about other ways that AI might be used in a related way. One question is, how is AI being used to understand the variety of immune responses to infectious disease? So it's kind of looking more on the host end of things, if you have any comments there. And the other is, in addition or apart from searching for new antibiotics using AI, can AI break down the genetic code of resistant organisms, creating molecules in real time that could exploit their weaknesses and eliminate the resistant bacteria?
Do you guys want to take the immunology one?
I know that there are some good case studies about, say with COVID, looking at like particular gene set responses that were predictive of like very poor COVID outcomes. So, with any of these AIs, essentially what you're doing is like complex statistics, right? Multivariate analysis. And how do you combine many variables and weigh each of them to evaluate on one criteria that's like your final output? And that's essentially what gene response is. So you can use the host cells as kind of like a molecular reporter of what's going on, right? So gene expression profiles are a great indicator of, the system, how the system's responding. So in the case of COVID, it was things like, if your epithelial cells are undergoing apoptosis at a very kind of high rate, or the gene profile for that was very high, that was one of the worst predictors of patient outcome. And, you could say that's obvious, but I think what's not obvious is that every single pathogen, right, we're talking about a complex biological space. And so the genes are responding to like this entire environment that's happening, right? Like how much death is it? What is the rate of death? What are the PAMPs that are associated with it? And all of that's integrated, it produces an RNA output. And then you can actually distill down, okay, which set of genes are specific to this type of molecular response that's predictive, say, for COVID outcomes.
Got it. Dionne, I'm even wondering how some of this might apply in a group way, like in congregate settings, or, if there are infection patterns where AI could come into play to, bring protocols into action. I don't know if these are things that are being talked about.
Well, I mean, AI is certainly widely used in understanding how infections spread, from like a computational sort of like external view. As for how they spread, looking at like molecular level, why, is it that COVID spreads like this and measles spreads like that? Maybe there could be machine learning that, does a deep dive into the DNA and RNA of these these viruses and figures out, well, okay, like right now we have, like let's say H5N1 becomes a thing. Maybe somebody could do a deep dive into the virus and figure out, is it likely to be, very high reproduction number or not? Can we in some way introduce some sort of vaccine, prophylaxis, something, to the population such that if you do get it, your body's able to at least maybe neutralize the ability for it to spread so easily.
Got it.
Some way.
And of course, that was the conversation that was going on about wastewater as well, where you might be able to leverage all of that data if we were still getting that data.
Yeah.
Anyway, that's a whole other story.
But actually, related to this,
Yeah.
like maybe the question is a little bit backwards, right? Like we should be considering this from like a patient perspective in the sense of, when a patient is infected by a bacterial or viral agent, right? They have a certain health status, right? And they have pre-existing conditions. And really, like, how do we use that type of information and integrate it? Like, how much do you weigh obesity and BMI? And how much do you weigh arthritis in this equation of like, what's your outcome going to be, right? So physicians, that's what we do, right? We're complex pattern recognition, and we make a prognosis. You see individuals and you respond to that. So maybe AI is like I don't like using AI, but like a way which we can integrate complex variables like this and actually gain more meaningful insights on outcomes. I think there's probably more promise there than just looking at purely at the molecular state.
Right. Interesting.
Going into the predicting future pandemics, we do have an interesting project in my lab where we're using recommendation systems, kind of like what Netflix uses to, predict if you've watched The Wire, you might like Breaking Bad or something. So we're looking at protein-protein interactions between pathogens, viruses, bacteria, etc., and their potential binders on host proteins on the surface of the host. And so, if SARS-CoV-2, we know that the spike protein binds to ACE2 on our cells, what will the next major virus, where will it bind? What are the main places where it might bind? And, we're using recommendation systems for that, with the ultimate goal of then being able to predict that and then being able to design molecules that can interfere with those, with the binding between the pathogen and our own cells. So I thought that's something that it's a very exploratory project still, but it aligns with the goal of thinking about future pandemics and trying to come up with countermeasures for them.
Got it. Let's see if we can fit in 2 more quick questions. We've got one right here.
Okay. Thanks really for this fascinating talk and panel discussion. So I had 2 quick questions. So one is related to, say, this approach of looking into the past and extinct organisms. Do you think that this has this, really interesting approach has applications in, say, in the detection of, say, anti-cancer, compounds or naturally occurring compounds to fight other, conditions or antagonize other conditions? And then the other question I had was related to combination of agents to treat or address antimicrobial resistance. So do you do that kind of modeling to, look at addressing AMR?
Yeah, thank you for the kind words and for the questions. I think so. I think by looking at all throughout evolution, we can find molecules for AMR or for maybe other problems that we have. So we're starting a number of collaborations to look at other properties. We have ourselves looked at immunomodulatory properties of a lot of these extinct molecules, and they have an ability to modulate the immune response of human cells.
Like
so
for
I think
autoimmune conditions?
Sorry?
You mean like for autoimmune conditions as well?
We just look at, we do multiplex ELISA, we look at ability to modulate cytokines and things like that.
Okay.
We haven't applied it to a particular disease yet, but they're able to modulate the immune response. Some of them have anti-inflammatory properties. So I think we're just at the very beginning of exploring all these new biological information, biological molecules, and that I think may be applicable to other problems, right, that we have. And the second question was
related to combination.
Oh, combination. Yeah, we've developed a machine learning model for synergistic interactions to predict them. It's still not very good. We did our first set of experimental validations, and it's not very good at it yet. But the idea is to predict interactions between different peptides and also peptide and small molecule antibiotics to
Yes.
be able to use them as adjuvant therapies and so on. But we're not quite there. It's not very good yet.
Okay, very good, thank you.
Thanks.
Excellent, thank you. And time for one last question.
Good morning. Thank you for all of this great discussion. Alison Fox-Robichaud, I'm the Scientific Director of Sepsis Canada. And as we've heard early this morning, it's a problem that we face in the ICU all the time in terms of antimicrobial resistance. I'm reading a fascinating book right now called Pathogenesis, and talking about how migration and individual pathogens have determined where we are in history, and how that has led to extinction of, prehistoric humans and others along the journey. We are a culture that migrates frequently now. There is a push against that. But as we think about how we migrate around the world, how do we assure that some of what's coming out of the AI platforms and others hasn't already been seen by our microbes and already considered that we have a solution to fight that because of the way that the current antimicrobial or the current microbes have evolved over history? I don't disagree with looking at the microbiome. I think it's going to be probably our most powerful tool, but we've got to change a little bit how we integrate that with equity issues, with everything else that's happening in the world with migration.
Great question.
Great question. I mean, one thing, I would align with, of course, infectious diseases are probably the top driver of natural selection, right, throughout evolution. So I think, a lot of our genes and the mutations that have occurred is due to the pressure exerted by infectious diseases that have decimated human populations all throughout history. So I think there's a lot to scratch there and to try to figure out, right? Currently, of course, the fact it is a hypothesis that present-day pathogens have not seen some of these molecules, but it might not be fully correct, right? Maybe some of them have seen similar molecules throughout evolution. We don't know that for a fact. So that's something to continue to explore. I think, those are my 2 cents on that.
Absolutely, and a good reminder that this is still a global issue,
of course,
requiring a global solution and everything that entails, including the issues of equity and distribution and who will benefit from these technologies. I feel like we could have another panel about this. Unfortunately, we are running out of time, so I'm now going to thank very much our panelists, Artem, Babaian, Dionne, Aleman, and of course our keynote speaker, César de la Fuente. Please join me again in saying thank you for their time. Thanks to you all here in the room and online for your fantastic questions. And I'm now going to turn it back to Mamatha. And I guess we can all. That should be. We'll stay. I guess we get to stay here.
Yeah. Thanks so much for this really fascinating talk and especially our wonderful panel. César de la Fuente, Dionne Aleman, Artem Babaian, and today's moderator, Ivan Semeniuk. Once again, this event is CPD accredited for physicians. To obtain your CPD credit, please scan the QR code on the screen and complete the evaluation form. We actually have another guest lecture coming up on December 10th at T-CAIREM with Dr. Tav Pritesh Shetty, who is an expert, an AI scientist and computational biologist who does work on infectious disease and global health. So this would be of interest, I think, to this audience. So please take a look at tcairem.utoronto.ca for further information. For the trainees who've registered for the meet and greet lunch with our panelists, we'll be heading over to the Forest Hill Room. And with that, I'll turn the microphone back over to Natasha Christie-Holmes.
Thanks, everyone. And you've already given the first instruction, which is for the trainees that have registered for lunch. If you head back to the registration desk, there'll be directions to the Forest Hill Room. For all other attendees at the conference, lunch will be provided out on the concourse. And another little reminder that bioMérieux has their traveling roadshow outside, their bus that you can check out all of their diagnostic kit out there. And just a big thank you to all of the panelists, to our keynote, to our wonderful moderator, Ivan. We're looking forward to resuming sessions here in this room and online at 1 o'clock after lunch. Thanks, everyone.